This is module 2.3.1 of the online course on genetic selection from concepts to application to genetic improvement of health. The topic of this module is quantitative trait genetics. My name is Jack Dekkers from Iowa State University, and I will be the presenter for this module, in which we will cover the genetic model that is typically used to describe phenotypes for a quantitative trait. We'll also talk about the concept of heritability of quantitative traits, the concepts of breeding values of animals for a quantitative trait, the concept of phenotypic and genetic correlations between traits, and how we can simultaneously select for more than one trait in the population. To target your learning, I'd like you to focus on the following questions as we go through this material. First of all, what does the formula P equals G plus E stand for? Second, what is the heritability of a trait? Third, what is the breeding value and why is it important? Fourth, how can breeding values be estimated? Fifth, what is the main cause of a genetic correlation between two traits and how does it differ from a phenotypic correlation? And finally, how can we simultaneously improve more than one trait? In this module, we will focus on quantitative traits, which include many of the traits that we're interested in livestock, including many disease traits, even those that are recorded in a discrete manner, for example, sick versus healthy. And the distinction between these Mendelian and quantitative or complex traits we discussed in module 223, with quantitative traits being affected by multiple to many genes or QTL, along with external or environmental factors. In module 223, we also discussed the effect of individual QTL on phenotype, depending on the genotype that the individual has at that QTL, and the average effect of alleles, which is based on the regression of phenotype on the number of copies of a given allele that individual carries. The standard model that is used in quantitative genetics to describe an observed phenotype is that it is the sum of genetic and environmental effects, or P equals G plus E. Here G represents the collective effect of the genotype that the individual has at all the QTL that affect this trait, and E is the collective effect of all environmental factors. For a population of individuals, we find that phenotypes often follow a bell shape or normal distribution, or phenotypes can be transformed to be normal. The phenotypes in a population are then characterized by the mean and the standard deviation or variance of the phenotypes. Note that this phenotypic variance is the result of variance in genetic values among individuals, in other words, the variance of G, and variance of environmental effects or the variance of E. Assuming that genetic and environmental effects are independent, this results in the variance of phenotypes or the phenotypic variance to be the sum of the variance of genetic values and the variance of environmental effects. The importance of genetics in explaining differences in phenotype between animals is known as heritability, and is defined as the proportion of phenotypic variance that is due to genetics, or the ratio of genetic variance over phenotypic variance. All these variances are defined within a herd. Thus, heritability is the proportion of differences among individuals within a herd that are the result of differences in genetics. This diagram further illustrates the concept of heritability. So if two individuals differ in phenotype by 200 units, for example for body weight at a given age, and this trait has a heritability of 0.25, then on average 25 of this difference that we see between individuals in phenotypes is caused by differences in the genetics between those individuals. So the expected difference in the breeding value between those individuals is equal to 25% of 200, which is equal to 50. It is important to know that this is only true on average across many individuals. So in reality, these two particular individuals could be more similar in their genetics, and more than 75% of the difference in phenotype we see may be due to environmental factors for this pair of individuals. But on average, 25% of the differences we see between individuals within a herd are due to genetics for this trait with a heritability of 25%. The table on the left shows some typical estimates of heritability for traits of relevance in pig production. So traits related to growth rate typically have moderate heritability, around 25 to 30%. Carcass composition traits typically have higher heritability, around 50%. And traits related to reproduction, disease, and survival typically have low heritability, for example, 10% for litter size and 5% for survival to weaning. So a large proportion of the differences in litter size and survival among pigs in a herd are the result of environmental factors rather than genetic factors. Trait heritabilities are important because they relate to how effective selection on phenotype can be to make genetic improvement. For example, if we select individuals for breeding that have the highest phenotype for a trait with low heritability, we expect only a small proportion of how much better these animals are in their phenotype to be due to genetics and transfer to the next generation. An important concept here is the breeding value of an individual, which relates to the genotype of an individual for QTL and average allele substitution effects at those QTL, as we've illustrated earlier. So for a single QTL, the breeding value of an animal represents the value of alleles that that animal passes on to progeny. Now for a single QTL, that breeding value for individuals for that QTL is the number of copies of a particular allele that the individual carries times the allele substitution effect. So in this example, the breeding value of a little q individual is 0 times 5, which is the allele substitution effect, which is equal to 0. The breeding value of a heterozygous individual, which has one big Q allele, is 1 times 5, equal to 5. And the breeding value of a big Q individual is 2, which is the number of big Q alleles it carries, times 5, the substitution effect, which is equal to 10. Now for traits that are affected by multiple QTL, we can get the overall breeding value of an individual as the sum of the breeding values that QTL that individual has. So for example, if the value of substituting a small letter allele by a capital allele is plus 1, so the allele substitution effect is plus 1, then the breeding value at a given QTL is just the number of capital alleles that the individual carries at that QTL, and the overall breeding value is just the total number of capital alleles, so we can get it just by counting for simplicity in this example. So then the breeding value of the sire in this example is 8 because he carries 8 capital alleles, and the breeding value of the dam is 7 because she carries 7 capital alleles. Now alleles are passed on to gametes after meiosis, so the genetic value of individual gametes in this example is also equal to the number of capital alleles that that gamete carries. And this can vary between gametes that are produced by the same individual as illustrated here. But on average, the genetic value of gametes produced by an individual is equal to half the breeding value of the parent, because the parent passes on half of the alleles. So 4 for the sire's gametes and 3.5 for the dam's gametes. The breeding value of individual progeny then is the sum of the genetic value of the parental gametes that combine to produce that progeny. So in this example, the breeding value of the first progeny is 4 plus 3 is 7, and the breeding value of the second progeny is 2 plus 4 is 6. This also illustrates an important concept, which is that progeny of the same parents don't have the same genetic value, because each progeny receives a random set of alleles from each parent. And these differences between progeny of the same parent are referred to as Mendelian sampling terms. The expected or average breeding value of progeny, however, is equal to the sum of the average genetic value of the parental gametes, so it's equal to half times the breeding value of the sire plus half times the breeding value of the dam, which in this case is 4 plus 3.5 is 7.5. And this is probably the most important result on this slide, which is that the average breeding value of the progeny is equal to the average of the breeding values of the sire and the dam. These average breeding values directly translate into differences in phenotype because phenotype is the sum of genetics and environment. Thus, then the expected phenotype of progeny is the average breeding value of its parents plus the overall mean. This also gives us a way to get the breeding value of a boar, which can be computed as 2 times the difference between the average phenotype of that individual's progeny and the overall mean, at least when that boar is mated to a random group of sows. Now, the factor 2 here comes from the fact that progeny only receive half the breeding value from the sire and the other half from the dam. This also illustrates the importance of selecting animals that have high breeding values for breeding, for producing the next generation, because parents with high breeding values tend to produce progeny that have higher phenotypes. And this is the main concept behind genetic improvement. So we try to select animals that have high breeding values and then use them to breed the next generation. The problem, however, is that we never know the true breeding value of individuals, but we can estimate breeding values based on phenotypic information, which includes own phenotype as well as phenotypes of relatives. And if we have genetic markers, we can also use genetic markers, and we'll talk about that later. Now, own phenotype provides information because animals with high breeding values tend to have high phenotypes, at least if that trait is heritable. The phenotype of relatives, however, also provides information on the breeding value of an individual because relatives share alleles. For example, if an individual has progeny with high phenotypes, then it's likely that the individual has passed on good alleles to its progeny and therefore has a high breeding value. Also, if the full sips of an individual have high phenotypes and therefore likely high breeding values, then the individual likely also has a high breeding value because the individual shares alleles with its full sip. In practice, all these types of information can be used to estimate breeding values, and animal breeders have developed statistical machinery referred to as animal model BLUP, or best linear unbiased prediction, that optimally utilizes all this information to estimate breeding values of individuals in a population as accurately as possible. And these EBV can be used to select the best sires and the best dams for breeding, because progeny of parents with high EBV tend to have high breeding values and therefore tend to have high phenotypes, as we've seen before. So animal breeders routinely compute EBV on all individuals in a population using all phenotypic data and all pedigree that's available. In swine, EBV is the common term used for these evaluations, but in other species other terms are used. For example, in beef cattle, the term that is used is expected progeny difference, or EPD, and an EPD of an individual is equal to half of its EBV, reflecting that progeny only receive half of the breeding value of the parent. In dairy cattle, the term that's often used is PTA, or predicted transmitting ability, and this is also equal to half of the EBV of the individual, and PTA quantifies through its name what is transmitted to progeny, which again is half the breeding value. EBV, EPD, and PTA are the most important tools that animal breeders have to make genetic progress. They are routinely obtained on animals in a breeding population for each trait of interest, and selecting the best animals for breeding then results in a progeny generation that has a better average phenotype than the parental generation, and that reflects genetic improvement. Selecting in the progeny generation to select the best animals based on EBV to produce the grand progeny generation then results in a grand progeny generation that again is better than the progeny generation, resulting in continuous genetic improvement. This has been very successful in many livestock populations, as is illustrated here, for example, for days to reach 250 pounds in the US Duroc population, which shows a steady decline in the average genetic value since about 1997. And this has resulted in pigs that now reach a higher market weight on substantially less feed, as is illustrated here on the left. However, selecting for growth not only results in genetic change in growth, but also in any other trait that has a genetic correlation with growth. For example, back fat has a positive genetic correlation with growth rate, so selecting for growth also tends to increase back fat. Genetic correlations represent correlations between breeding values. So the genetic correlation between growth and back fat is equal to the correlation of the true breeding values of individuals in a population for growth with their true breeding values for back fat. Genetic correlations are caused by QTL that affect both traits, which are referred to as pleiotropic QTL. So when considering all QTL that contribute to the breeding value for trait 1 and all QTL that contribute to the breeding value for trait 2, as indicated here, there is a genetic correlation if some QTL affect both traits. Genetic correlations are different from phenotypic correlations, which are correlations between phenotypes observed for the two traits among individuals in a population. The phenotypic correlation between two traits is a combination of the genetic correlation between traits and the environmental correlation between traits. Environmental correlations between traits are caused by environmental factors that affect both traits. These tables give examples of estimates of genetic correlations among pigs. For example, feed conversion has a negative genetic correlation with average daily gain, but a positive genetic correlation with back fat. So selection for growth rate will not only improve growth rate, but it will also reduce or improve feed conversion rate as a result of this negative genetic correlation. In practice, breeders never select on a single trait, but multiple traits are selected on simultaneously. For example, we not only want to improve growth rate, but also reduce back fat. So we have to select simultaneously for increasing growth rate and reducing back fat. One way to accomplish this is to practice what is called independent culling selection. This involves setting a threshold for the EBV for growth rate and a threshold for the EBV for back fat, and then only use animals for breeding that exceed the threshold for growth and that are below the threshold for back fat. And this is illustrated in the graph, and the cloud of points here reflects the positive correlation between EBV for growth and back fat, consistent with the positive genetic correlation between those traits. With the thresholds given by the blue lines, animals that fall in the lower right quadrant are the ones that we select for breeding based on this strategy. Now note that selected animals have above average EBV for growth rate and below average EBV for back fat. So if we use those animals for breeding, we expect to increase growth rate in the next generation and to reduce back fat. Note that selection on growth only would have increased back fat because of the positive genetic correlation. An alternative and actually better way to select for multiple traits is to practice what is called index selection. This involves computing an index for each animal as a weighted sum of its EBV for the traits that are in the selection. Now, the weights here can represent the economic value of each trait, so the effect of increasing that trait by 1 unit on profit. So in this example, the economic value of growth would be positive and that of back fat would be negative. And animals selected for breeding are then the ones whose index exceeds a certain threshold. For this example, this is represented by the individuals that fall below the red line in this figure. From this illustration, it is clear that these two strategies do not select the same animals. Specifically, index selection allows animals with EBV for growth that are below the independent threshold for growth to be selected if their EBV for fat makes up for that difference. Now, it's been shown that index selection results in a greater response to selection in profit, which of course is the ultimate goal. So index selection is what is typically used and recommended when practicing multiple trait selection. And also index selection can easily be extended to include more traits by just adding that as the weighted sum. This concludes this module, and I now want to leave you with the questions that I referred to at the beginning of this module. So what does the formula P equals G plus E stand for? What is the heritability of a trait? What does it refer to? And how is it interpreted? What is a breeding value and why is a breeding value important? How can breeding values be estimated? And what is the main cause of a genetic correlation between two traits and how does that differ from the phenotypic correlation between traits? And finally, in what ways can we simultaneously improve more than one trait? Thank you for your attention.